want to thank the organizers for bringing us together. And let's just get started. So, Tom, I'll start with you. Why don't you kind of give us your perspective on, state-of-the-art gene augmentation or manipulation and RNA strategies and kind of where we are today with it?
Sure. Before we talk about gene manipulation and RNA modulation, maybe backpedal a bit and talk about straightforward gene augmentation. I was at Spark previously, and gene augmentation is usually used to replace a gene, to add a gene to a cell. And it works best for autosomal recessive disorders like RP65-mediated IRD. But the problem arises when the gene is too big to fit in traditional AAV2 vectors, or there's an autosomal dominant disease with a dominant negative gene. And so for those indications, you need to potentially knock down and replace a dominant negative gene, or you can use these other modalities that you mentioned. And one modality that's interesting is RNA modulation. So the leader in that field was ProQR. They manufacture ASOs or oligonucleotides. And these are really interesting because they're administered in the office. They don't involve a viral vector. There's less propensity for inflammation because they don't involve a viral vector. And because they're administered intravitreally, you can treat the entire retina and potentially modulate gene expression of the entire retina. And so for large gene disorders, this may be a path forward. So ProQR had a phase 1 study for LCA10, which is mediated by the CEP290 gene. It's a gene that's too large to fit into AAV. And this original study looked really promising. And when they did the follow-up phase 2/3 study, unfortunately, it didn't meet its primary endpoint. They were also looking at other large gene disorders, Stargardt and Usher, and I think autosomal dominant RP, which would have involved a knockdown and replace. But in any case, it didn't meet its primary endpoint, unfortunately, and they decided to, I think, sell off their eye franchise. So that's sort of in limbo now. And then you mentioned gene editing. So the leader in that field has been Editas, and they were also working on LCA10, the CEP290 gene, and they recently announced that study, it was a phase 1 study, didn't meet its primary endpoint. And so, I'm actually really concerned as a retina specialist and somebody who's devoted their career to advancing the field forward, that we've had these failures recently in gene therapy. We've also had some high-profile failures in choroideremia. And, I think this is a great forum to potentially discuss, what happens if we keep having failures in gene therapy and ophthalmology. You wonder, will investment dollars continue to flow into our field? What's the future hold? And so I'm really hoping that we have some successes, some high-profile successes in gene therapy, so we can continue the flow of funds into our field to fund research and development. And I'm really excited about some of these therapies. The Adverum therapy is very exciting because it's a very important disorder. AMD is the leading cause of blindness. REGENXBIO is working on that as well. And so I'm really hopeful that one of these therapies eventually is approved. So I think it will really enhance the flow of funds into our field and continue advancement into our field. So, maybe after we can have a discussion about, how do we continue to attract those investment dollars into our field?
Yeah. Thank you for that. I think we could have a whole session, a day-long session just on that topic. So I'm going to shift over to you, Dr. Wiley, as, just based on what Tom just said. I was thinking, what have the field learned about clinical trial design? But I think we're learning about considerations for clinical trial design that is going to inform endpoints and novel endpoints. You want to comment a bit on that?
Absolutely. And so first, I'll thank the organizers for the invitation. This has been my first chance to attend an OIS event, and it's a wonderful forum. I'll pick up where Tom left off. And so if there was a huge wave of excitement surrounding the approval of Luxturna back in 2017. Then we've seen some disappointment since then. And I think we're all asking the question about whether this is a problem with the biology and our therapeutics or limitations to our clinical trials. So the answers are only as good as our questions. And for this, I'll talk a little bit in reference to the FDA's Guidance for Industry 2020 document regarding human gene therapy for retinal disorders. So we're fortunate to have very explicit guidance or advice from the agency on retinal disorders in this realm. To my knowledge, there's no corresponding advice or guidance document regarding cell therapy. So that is specific to the retinal disorder realm. So the agency will sometimes reference the gene therapy guidance on considerations related to cell therapy, which we'll talk a little bit about as well. And the section 4 of that document contains considerations for clinical trials. And there isn't time to talk about a lot of those bullets, but I'll mention a couple. There is consideration for controls and for adequate masking as means of trying to minimize bias in a trial design. And there are challenges here. So the agency recommends that we use vehicle administration where possible as a placebo control. And obviously our delivery methods for these therapeutics in the retina space are invasive enough. The agency acknowledges that there has to be judgment made about whether a vehicle control is really suitable. So a lot of our trials end up employing sham controls or non-treated controls instead. There are additional considerations to try to get around the bias that's sometimes injected into a design in that regard. So the agency gives advice about trying to use alternate dose regimens. But there are challenges there too. So for gene therapy, for instance, we see sometimes dose-limiting toxicity, for example, in the form of inflammation with some of our gene vectors. And the therapeutic window may therefore be somewhat narrow. In the cell therapy realm, it's not clear whether for some of these therapeutics you'd expect a normal dose-response relationship. Think of what it is to do tissue replacement, say with an RPE cell therapy, and would you expect a different outcome depending on the precise number of cells that you were injecting? On the masking front, similarly, for our delivery procedures, there are limitations to how well we can mask patients. And this is particularly true for some of the delivery methods that require surgery in the operating room. And then to speak to clinical trial outcomes, in this document, the FDA explicitly mentions 2 that are approved efficacy outcomes in this space. They reference best corrected visual acuity, where, as we talk about either halving or doubling of the visual angles, so a change of 15 letters on an eye chart. And this we know is a challenge, whether because in many diseases this is not a measurable endpoint over a feasible time horizon for a clinical study, or because other parameters of visual function are more relevant or operative. Separately, they mention the example of rates of photoreceptor loss on imaging modalities such as fundus autofluorescence and optical coherence tomography. And this has been a real bright spot. There have been successful clinical programs that have taken advantage of this expansion of clinical trial outcomes in recent years. But there are limitations here too. So think of X-linked retinoschisis, where the prototypical cavitations within the retina don't allow you to make an assessment of the outer lamination of the retina by OCT, or consider a cell therapy like injection of RPE subretinally or insertion of a subretinal RPE patch, where the insertion of that RPE or injection of that RPE is going to change the hyperreflective lamination of the outer retina and make that ellipsoid zone measurement more challenging. So the FDA encourages sponsors to engage with the agency about development of novel endpoints, and they specifically use the example of the multi-luminance mobility test that was developed in the context of what eventually became successful efficacy claims for Luxturna. And that's both resource intensive because this often requires what amounts to a separate study before you start clinical development, specific to a disease area where you want to seek validation for your measure before you enter clinical trials. And it's probably impractical to carry that out over many diseases. And so I think all of these represent some challenges. Nice to have the explicit guidance that we do. I think in the end, we're seeing enough bright spots to realize that there are limitations to our clinical trial design that I think we'll overcome. And I think the fault is often not in the biology or our therapeutics. I think it's often the way we're able to measure things. We'll talk a little bit more perhaps about some of this. But clearly it's been a challenging time for a number of programs in recent years. And I'm looking forward to what Tom said, I hope for some successes so we can turn this around.
Yeah, I just wanted to add to that, just to emphasize. So when I was at Spark, the MLMT, multiluminance mobility test, was sort of popularized. And most of my colleagues are very surprised that, as you said, it was a trial in of itself. It was a year-long trial to validate that. And oftentimes these endpoints need to be validated specifically for the disorder. So it can be, as you said, very labor intensive. And I think that's a challenge. And I think the other challenge, getting back to why we've had some failures, I think particularly for IRDs, we may be enrolling patients in trials after their disease has gone too far. If they don't have much retina left, they have a lot of retina atrophy, it's very difficult to show a benefit in some of these patients. And I think, particularly with the choroidal anemia trials, we were intervening in those trials when patients had very little normal retina, or very no normal retina left, and very little functional retina left. So I think that's another challenge with clinical trial design is the eligibility criteria. So I think alternative delivery methods that don't involve subretinal surgery, a trip to the operating room, are really important because this would allow us potentially to enroll patients with earlier forms of the disease and maybe realize a benefit. Because to your point, I think the biology works. It's just a matter of are you enrolling the right patients? Do you have the right endpoints? Another surprise with my colleagues, I remind them that Luxturna did not hit on statistical significance for VA. So, as ophthalmologists, we're sort of addicted to VA, but it's the wrong endpoint. And so these other endpoints are super important.
Agreed.
If I may.
Yeah, please.
Since we started the discussion. So I would like to comment on the endpoints and the new endpoints because there is a lot of discussion about new endpoints. So I wanted to remind people that for the Luxturna, the MLMT started after the first administration to patients. So it's not necessary to be all afraid of we have to have it in advance before we really start going into patients, but it has to be there before the phase 3 trial because the phase 3 is confirmatory. So that's where the interplay of how we can move fast, but at the same time account for all the necessary work in parallel. And to highlight also the relevance of the most appropriate endpoint for the patient experience. So I think as a field, we need to be brave and also to be considerate of what the patient feels it is valuable to them. And it may not be the visual acuity that we are all used to, as Tom just said, but also take into account what do we want to do is to bend the natural history of the disease. So that's where the natural history studies become very important. I wanted to just remind people of that and to also identify from the patient's experience what's the most relevant. I had experience where we actually had in another rare disease or variety of retinitis pigmentosa to use the delay in dark adaptation, and the regulators accepted that this could be a valid endpoint as long as we substantiate in this specific patient population.
Well, Dr. Stacy, while you're talking, why don't let's bring cell-based therapies into this conversation because they're not excluded from this conversation. I mean, we've learned a lot. Yesterday, we heard from InGel, and they have a great cell reprogramming as a neuroprotection. So, can you speak a little bit about cell-based therapies and?
Yeah,
how
no,
that
thank
fits
you.
in.
So, cell-based therapies, I'm very excited about because they have a tremendous potential for both the front and the back of the eye. And despite considerable hurdles that should be ideally addressed as soon as possible, it is still a very, very promising solution for tons of indications. So in full disclosure, I'm probably biased because I have spent half a decade in the lab and a decade leading designing and executing cell therapy clinical trials in the both in the front and the back of the eye at Novartis, and last year at 10 Point Therapeutics in cell therapies in ophthalmology with the lead product on embryonic stem cell derived RPE in an ongoing phase 1/2 clinical trial in collaboration with Moorfields. So it's very near and dear to my heart. But I would like to remind people that both this huge potential as well as the potential hurdles, in my view, a lot of them stem from the same basic attribute of the cell therapies, which is that they are plastic, they have plasticity and adaptability into the interplay of both the intrinsic cues that the cells have, as well as their environment, whether that environment is in culture, when we try to manipulate them and manufacture them, or it is after they get into the human body. So some key points to consider. First of all, I think we all need to be very clear, and sometimes it's blended, whether we target cell replacement versus trophic support. And I have to say from my experience, especially in Novartis, in due diligence of a lot of things in the field, pharma leaders especially, they get very nervous when they start hearing about trophic support because they are very used to low molecular weight compounds where it's extremely defined what we manufacture and we have really good control versus when we start talking about producing a whole lot of different trophic factors. But I think that there is a lot of potential there, and it's really critical, as we heard yesterday, to really start looking at a big panel of what these cells produce. The next point that I would like to remind people about is that one of the potential drawbacks is that currently we don't have a really good way of assessing the characteristics of the cells. So that makes especially manufacturing a little bit problematic, and how we are going to have a good in vitro potency, ideally, of these cells in order to especially be able to characterize what happens and evaluate what happens in clinical trials, whether we have potentially different batches going into different patients, and some of the patients respond and some of the patients don't respond. So that's where it starts becoming critical. And the last point I would like to touch on is delivery. So delivery in cell therapies, it's even more critical than other products because, number one, we want the cells obviously to be unharmed, but also in certain indications, it is very critical to take into account where these cells are going to go, and are they going to be able to integrate where they go, and plus, where they go, is it going to be already some remodeling, for example, of the retinal circuitry that we need to take into account in order to have a specific expectation of a treatment effect? So all this into account, I'm very excited to talk with anybody about cell therapies. Happy to be a sounding board. I'm switching right now my focus professionally into gene therapies. You hear that I'm a co-founder and scientific advisory board at Diurasis Therapeutics, which is a very early startup on gene therapy for glaucoma with targeting lowering of IOP. And just like a day ago, I joined the Adverum team working closely with Rick and Laurent. But I'm really happy to help anybody to move the cell therapy field as well along. I think it's huge potential.
Just for fun, I'm going to throw in a spicy comment. I'm not very excited about cell therapy. I think that it's really interesting, but I think it's very long term. I think the results that I've seen at meetings aren't very impressive. I think it's a way off. And I know investors love moonshots. I think it's a bit of a moonshot, but I think it's more of a long-term, goal for clinical trials. But I think there's a lot of work to do in terms of, we don't even know how to place the RPE cells in the eye physically. We talk about scaffolds and gels, and I just think there's issues with immune rejection, and I think it's obviously important to do, but I think it's a ways off.
So Tom, to that, one of the, really attractive features of cell-based therapy is that it's gene agnostic. So do you have some favorites, gene agnostic approaches?
Well, I think, yes, I think, something that's not quite a way off. I think optogenetics is gene agnostic. I think we heard a really great presentation. Nanoscope, with whom I advise, has some interesting results from their early study. They're currently in a phase 2 study. They'll have results next year. I think this is a really interesting gene agnostic approach. As we heard, you induce light activated ion channels in the retinal neurons, and patients can at least regain some ambulatory vision. I think neurotrophic factors, sparing vision's working on rod-derived cone viability factor, that has some potential. But I think that's much more near-term than cell therapy. And don't get me wrong, I hope cell therapy works. I just think it's a ways off.
Thank you. Anyone else?
I may add that I'm really excited about the things you mentioned. To remind people also that we can use the gene therapy as a cell factory. So I would encourage people to really look into that, because that can bypass the gene mutation limitation. As long as we have good pathways. And targets.
All right. So let's just talk about some very specific things. Delivery, dosing, inflammation, as it relates to these gene therapies. Have we gotten it right? What do we kind of need to continue to talk about to improve on that? Who wants to take that first?
I'll take it. So the traditional way to administer gene therapy is subretinally. We know the subretinal space is an immune privileged space, and you'll have much better tolerance. I'm always surprised when colleagues are surprised that there's inflammation after gene therapy. Our immune system has evolved over millions of years to fight off infections. You put 500 billion viral particles in somebody's eye, you should have inflammation. So I think it's really remarkable that we have these vectors with rational design and directed evolution that minimize the immune response. And certainly placing these vectors in the subretinal space makes a lot of sense to minimize immune response. But clearly that's not the path forward for common diseases. It might be for an ultra rare disease like RP65, but for common diseases like diabetic retinopathy or AMD, alternative office-based, less invasive means of administration are necessary. Hence the interest in suprachoroidal and intravitreal. But the problem obviously is those the intravitreal space and the suprachoroidal space aren't traditional immune privileged spaces. At Clearside, we did a study with Glenn Yu at their primate facility, and we compared AAV8 vector-carrying GFP administered subretinally, suprachoroidally, and intravitreally. And we found that when we administered suprachoroidally versus intravitreally, there was some inflammation locally, and the paper's published and it's freely downloadable. But basically, the bottom line was that the immune response was to the GFP being a very foreign protein and not to the vector. Intravitreally, there was some systemic antibody response. So it looks like, obviously, the subretinal space is best. The suprachoroidal space, I'm not going to say it's immune privileged, but it has some immune tolerance. The intravitreal space, maybe less so, but with these designer vectors, the 7M8 that Adverum's using seems to be well tolerated. So I think that's just a little bit of a primer on delivery. But I do think ultimately we need to move away from subretinal surgery that's invasive and fraught with complications.
Thank you.
Can I comment? So I totally agree with everything that Tom said. I just wanted to remind people a little bit of the history and what I had seen also in due diligence from Novartis, looking at pretty much everything out there, confidential data that I'm not going to discuss, but I can discuss the bottom line of that. So a lot of programs have seen inflammation dose-related in clinical trials, and they have not published the results. So that, especially with subretinal injection, because mostly that's what has been used so far. So we should not be surprised. Remember that if you look at the specific dose response that Luxturna looked at in the first clinical trials that they did not really push the dose really high because they were afraid of inflammation. My mentor was Jim Bennett, so I was at Penn when this study was happening. So on purpose and for good reasoning, absolutely good reasoning, they didn't want to push the dose up. So what we saw in Luxturna had a rationale. If we start pushing the dose up, it's not surprised that we are going to be seeing inflammation. And a lot of programs have seen much, much more intense inflammation than what has been published already. So we need to be vigilant, but not afraid of finding the right dose.
Thank you. And so with that, we're going to close this session. So join me in thanking these wonderful panelists for this discussion. And look forward to talking to you during the breaks. Thank you.
Thank you for having us.
Thanks.
Thank you.